Good morning or afternoon. My name is Janie Montblanc, and on behalf of the Great Basin Fire Science Exchange, the Native Plant Project, and our partners, I would like to welcome you to this webinar titled Selection of Genetically Appropriate Plant Materials for Increase, presented by Holly Prendeville with the Pacific Northwest Research Station. Before I introduce our speaker, I will go over some webinar details. If you have questions for the speaker or me, please type them into the questions pane of your control panel located at the top right of your screen at any time during the webinar. It helps us to receive questions throughout the webinar so that we have some lined up in the queue to begin addressing promptly after the presentation. I also want to let you know that whatever you do in your control panel does not affect the webinar presentation, so you're welcome to type a test message or test your audio at any time during the webinar. If you're having problems with your audio, please open your audio pane and check your audio selections. Now I would like to introduce our presenter. Holly Prendeville is a research geneticist for the U.S. Forest Service Pacific Northwest Research Station in Corvallis, Oregon. She has been a field biologist for almost 20 years. She obtained her PhD in biology with an emphasis in ecology, evolution, and behavior at the University of Nebraska, and completed a postdoc at the University of Virginia examining factors that affect population evolution in a forest herb in the eastern U.S. As a research geneticist with the Forest Service, she is examining the efficacy of seed zones for blue bunch wheatgrass in the Intermountain West. Welcome, Holly, and thank you for presenting today.
Thank you very much for the opportunity. Hello, everyone. Today I'm going to be talking about using seed zones to select genetically appropriate plant materials. Oh,
I just made you the presenter, yeah.
Yes, now you can see my screen.
Yes.
Excellent. So in the federal lands, we have directives that indicate that we need to use genetically appropriate materials that are adapted to the local environment. This is essential so that we can have healthy ecosystems and make very small impacts on the remnant native populations. However, this can be very challenging. In that, so sorry, can you see that the, my Dropbox,
I can't see, no, I see your,
okay,
just your slide.
Okay, okay, sorry, thank you.
That's okay.
So sorry, back to this is distracting me, obviously. So back to the seminar here. So obviously this is becoming increasingly challenging with more and more acres succumbing to wildfire, and with 2015 being a record-breaking year with over 10 million acres burning. So today my talk is broken up into three components. First, I'll go into describing seed zones and the various seed transfer guidelines that are available. Then I'll go into the tools that are available to make this easier. With all of the demands that are out there, having more tools to make this easier for you, it will improve our restoration efforts and improve our restoration efforts. And then finally, I'll talk about my research. Okay, so the National Seed Strategy was published in 2015 and lays out a number of goals and sub-goals. And in the first two goals, they indicate the use of genetically appropriate material, having them more readily available, and then also conducting more research so that we can improve the technology and improve our restoration efforts. So why has there been this emphasis to utilize genetically appropriate material? This is because we do see that plants are locally adapted. Even though a species itself may be very plastic, so I'm showing the example of big squirrel tail here, and it can occur in environments from 600 to over 3,000 meters in elevation, from desert to alpine ecosystems, as well as in a variety of soil types. So even though the species itself is very plastic, there can be adaptation at the population level. So this is work done by Courtney Rowe and Beth Ledger looking at Elmus multistatus or big squirrel tail and the percent emergence. And in this study, they took seed from 4 populations and grew it in this local location. And what I've done is added the minimum winter temperature as well as the annual heat moisture index or aridity index for each of these populations. And we can see that the percent emergence was the greatest for the local population, which was a population in California, and then less so for other populations in Oregon, Idaho, and another California population. one thing I'd like to note is that two of the populations had a similar minimum winter temperature as the local population, but differed in aridity. So this site is much drier, a little less so, and I should note that less than 2 is indicating a very moist environment. And as you increase in numbers, it's getting drier. So this may be the reason why we're seeing a difference, but this is clear evidence is that the local population is doing the best. So local adaptation occurs when a population evolves in response to selective pressures imposed by the local environment. These selective pressures can be climate itself, but also can be biotic factors such as competition from invasives or intense herbivory. Another example of local adaptation you may be familiar with is due to soil types, particularly extreme soil types by serpentine soils. Serpentine soils have heavy metals in them. And in this study, we see that populations in a similar climate were taken from different soil types and then reciprocally planted into the other soil types. So we see the serpentine soil has the highest number of seeds per plant when it's planted in serpentine soil. And then plants that came from non-serpentine soil also have the highest reproduction when planted in non-serpentine soil. So this is clear evidence of local adaptation due to the soil type. And all of this is indicating that there are genetic differences even on population levels. So hence why we need to look for genetically appropriate materials while we're doing restoration. I'm going to go into the different seed transfer guidelines that are currently present and explain those. First is ecoregions. Ecoregions were not developed to be a seed transfer zone specifically. They were designed to differentiate ecosystem quality and integrity based on the factors that are listed here. Ecoregions have 4 different levels. So level 1 is indicating very broad areas, and then it's increasing in levels of distinction. As here we are in level 4, just looking at the Washington and Oregon coast. And you can see there's a number of levels even within this small area. Though ecoregions were not designed to be seed transfer zones, they are commonly used as such because they are indicating differences in ecosystem quality and integrity. And we'll come back to these, particularly ecoregion level 3. Breeding zones. Breeding zones are designed for tree improvement, particularly for growth and sometimes also disease resistance and wood quality. These are areas in which a single population of improved trees or trees that have been selected upon to have these qualities can be planted without maladaptation, so they'll do well in those areas. Here are 2 maps of breeding zones. We have western red cedar and Douglas fir, looking at a similar area on the western Oregon. And you can see for western red cedar, it's broken up into 4 different breeding zones, whereas Douglas fir is up to 16 different breeding zones for the same area, indicating that these 2 species adapt differently and selection is acting upon them in different manners, resulting in different maps for these species. Seed zones. Seed zones are meant for native plants, wild populations, and not meant for selected lines that we have gone through to select for different traits, as with breeding zones. These are meant for your wild populations and going for a healthy ecosystem. This is plant material that will be genetically appropriate to the local environment and will allow us to maximize our restoration and reforestation efforts, as well as limit the negative impacts on the ecosystem health and the remnant native populations. Provisional seed zones were designed specifically as a seed transfer guideline. Provisional seed zones were also designed so that for species in which we have very little information about and don't have empirical seed zones, which I'll describe here in a bit. Provisional seed zones take 2 climatic factors, minimum winter temperature and aridity, and combine those together. Minimum winter temperature should look quite familiar in that it is comparable to the plant hardiness guides that you're probably seeing in your nursery catalogs that you're receiving this time of year. And then aridity takes the annual mean temperature and divides that by the total precipitation, and the units for this is Celsius by meters of precipitation. And so the levels for this annual heat moisture index or aridity, the dark green is indicating a less than 2, so these are very moist areas as we have here in the Pacific Northwest and in the Northeast. And then as you increase in value, it's getting hotter and drier in these areas. And so using spatial statistics, these two factors are combined together to generate this colorful map. And so the idea with provisional seed zones is that if you stay within a color, you can transfer material within that area and it would be locally adapted to that climate. Suggested by the authors, which are Andy Bauer, Vicki Erickson, and Brad St. Clair, are to also incorporate level 3 ecoregions so that you are including that ecosystem quality and integrity. So rather than going throughout an entire color, you would actually stay within these black lines within the color and transfer seed among those areas. For the Great Basin, we have fewer ecoregions and actually more provisional seed zones, and so you can just stay within your ecoregion and determine the provisional seed zone for which you should transfer seed among. So how are these genetically appropriate? Well, the authors of this work took common garden data. So when we grow various populations at the same site, the differences we see among those populations are due to genetics. And so they took common garden data from different works and compared to see how these different zoning methods accounted for genetic variation. And so the green boxes are indicating a lot of genetic variation is accounted for these traits looked at. And these are traits important to survival and reproduction, grouped by phenology, morphology, and reproduction here. And then we have decreasing levels of genetic variation with an intermediate color, and then white indicating a very low amount of genetic variation is accounted for within this trait. And the different zoning regions that they looked at were the level 3 ecoregion, the provisional seed zone with the level 3 ecoregion, and the empirical seed zones, which I'll get to here shortly. One thing I'd like to point out is the number of zones for each of these zoning methods. So we're looking at the same area, but each of these zoning guidelines break that area up into different regions. So we can see that for some traits there's a comparable amount of genetic variation that's accounted for with just provisional seed zones alone, and in comparison to provisional seed zones plus level 3 ecoregions. And we do have more genetic variation accounted for using empirical seed zones. So here are two other studies which they compared, or two other species that they compared a variety of traits, and again found that the provisional seed zones along with the level 3 ecoregions and the empirical seed zones account for a lot of genetic variation. So this provisional seed zone guideline method does account for genetic variation and should then help you guide in where you can move seed so that you have populations or seed that is locally adapted to the environment you're restoring. In a separate study done by Andrea Kramer and her colleagues, and Andrea will be talking next in this webinar more about the genetics, so please tune in for that. She examined Aragonium species and Penstemon species, and again, similar context here with accounting for genetic variability, with the dark green indicating a lot of genetic variance accounted for, and the white indicating a smaller amount of genetic variance accounted for. And here she looked at the same zones, but there were no empirical seed zones for these species, but she looked at the level 4 ecoregion as well as the populations. And here we're seeing the range of zones available for each of these 5 species. And she also showed the R-squared values. So here the values are indicating the proportion of genetic variance that is accounted for by each of these zoning methods. So you can see for each of these zoning methods, population does account for the most genetic variance, but when it comes to the feasibility of having seed for every single population on all the millions of acres that we need to restore, that is just not financially feasible. So if we can reduce the number of areas that we have seed for, and that would be genetically appropriate, we don't lose very much genetic variance. And so these methods do work in accounting for genetic variance. And again, she looked at traits that were important to germination, survival, and reproduction. So again, these methods would be useful for getting locally adapted material. And finally, we have empirical seed zones. These are specific to a species. I'm going to describe how seed zones are made for a species. And so you have your region of interest. Ideally, and if financially possible, you'd make it for the entire distribution of the species. But that's not always possible. So then if you can get it for the range of climates that the species exist in, that would be useful. And then, of course, there's just the limitations of the area that you're interested in. So you would go to that area, you would collect populations throughout that area, and then plant them in a common garden site. Again, if they're planted in a common garden site, the differences we see among those populations are going to be differences due to their genetics or their interaction with the environment and the genes. And so, again, we'll look at traits that are important for survival and reproduction, such as morphology, phenology, and reproduction, and then take those data and using spatial statistics, combining it with the climatic variables from each of the wild populations you used in your study. And then you would generate a colorful map. And again, you can then incorporate the level 3 ecoregions to incorporate differences in ecosystem quality and integrity. So that's the basis of how most empirical seed zones have been developed, particularly the more recent ones. There have been a number of empirical seed zones that have been made for species throughout the United States. I'm just emphasizing some that have been made for the western United States, and also want to note that these have been made for plants in a variety of different plant families. So as a land manager, you want to restore area. How do you go about this? So first you're going to figure out the species that you're working with and see if there is an empirical seed zone map for the area that you're working in and for the species that you're working with. Also, if you're working with reforestation, breeding zones will work well. If neither of these are available for the species that you're working with, then you'll need to move to the provisional seed zone and determine what seed zone you're in and then what level 3 ecoregion that you are in. With that information, you'll know from which areas you're going to be able to obtain seed to restore your area. And then, of course, last and definitely not least, but is your local knowledge of the area. If you know you're going to restore an area with serpentine soils or mine tailings, then or that has lots of invasive species, you can utilize that knowledge and see if you can get seed that is adapted for those issues. So you definitely want to incorporate your knowledge of the area, and that these provisional seed zones are a guide, and then your knowledge will help improve the restoration. And of course, also ask for help. So your geneticists, your botanists, or the authors that made the seed zones, you should reach out to them. So here I'm listing the state botanists with the BLM, and then the regional geneticists with the Forest Service. This information is available to you, as well as all of the URLs in the handout associated with this webinar, and I'll come back to it if you need to see it again. So now the tools that are available. The Western Wildland Environmental Threat Assessment Center, or WWETAC, has a variety of tools and links to them, links to a variety of tools. And so one of the tools they have is the Seed Zone Mapper. The Seed Zone Mapper is really useful. It has all the shape files, so if you use ArcGIS, you can download the shape files and then modify them as you need. There's also PDFs of the seed zone maps, and then the published work is accessible through here as well. And so then you can get the contact information for the authors and reach out to them if you need assistance with figuring out your seed zone. Patrick Kraybacher, a technician I'm working with, and I have been working on converting all of the seed zones that are available on WWETAC's site into Google Maps. And here are the seed zones we have. So we were able to convert the provisional seed zones, and we had to break them up into regions of the Forest Service. That's indicated here what each of these regions are, as well as we were able to make one for the Great Basin itself. And you can reach these through links that are provided on the handout as well as on WWETAC's website. In addition, for the empirical seed zones that were available on WWETAC, we also converted those so that they're able to be viewed on Google Maps. These maps you can also access on your phone through the Google Maps app. And if you have a Gmail account, you can then save it for future use and modification. This is the Great Basin Provisional Seed Zone map. I will note some of the colors may be different from the publication colors because we're limited by our choices with Google Maps. But you'll be able to see the legend here. So we have the minimum winter temperature and the aridity for each of these seed zones. It has all the functionality of Google Maps, so you can zoom in into an area. So here we're looking at Winnemucca, and you can see the variety of seed zones, provisional seed zones that are in and around this area. And you can use your mouse and click on a polygon or a particular color, and a label should pop up giving you the minimum winter temperature and aridity or annual heat moisture index, and give you that value. So you'll know what provisional seed zone that you'll be in, and then this way you don't have to figure out what shade of yellow you're working with either. Here we have the provisional seed zone map for region 4. Again, the color coding isn't always consistent among maps, and why I suggest just clicking on a seed zone so you can determine which seed zone you're within. But if you scroll down at the bottom, you can see that the level 3 ecoregions are present here. You'll need to click on these to highlight them, and because these are layers, once they're highlighted, you can't highlight the provisional seed zone map, but you can then see what ecoregion you're working within, and then even click on it to highlight to get the name of it as well. Here we have an example of one of the empirical seed maps. This is Blue Bunch wheatgrass. And again, if you have a Gmail account, you can save these maps and make modifications. And when you make modifications, you won't change the map that's accessible to everyone. You don't have permission to edit that one, but once you save it to your account, you have permission to then do whatever you want with it. So here you can indicate, say, locations where you've noted wild populations or made seed collections, or in my case, I'm indicating where my common garden experiments are located throughout the seed zones. In addition, what else is available on WETAC is that there's going to be a mobile app that will be coming out soon. This mobile app does have offline functionality, whereas our Google Maps, unfortunately, you can't save the layers for an offline functionality, but if you do have it on your cell phone and you have cell phone service, then they will be accessible. So just to give you an idea that this provisional seed zone map is in the works and will be coming soon via collector for ArcGIS. As I said, it has functions offline. You can download the maps for your area at a variety of scales, as well as make notes, look at the coordinates, and note coordinates of sites. And soon it will have the Seeds of Success collection form. So here's just a quick glimpse of what it currently looks like, showing you different colors for the seed zones that you'll be able to click on to indicate what seed zone you're in, as well as you'll be able to hit this plus sign to then add a location. So you can add a location of the seeds you're collecting or where a population exists. And this form will look like the Seeds of Success data collection form. In addition is the Seed Zone GeoBrowser. This is a fantastic tool to use on your desktop. It has both the provisional and empirical seed zone maps. You can also put in lat-long to figure out where you are and what region or what provisional zone you're working in and empirical zone you're working in. You can also view different climate change layers and a variety of boundary layers. And this tool will allow you to generate PDFs as well as some other JPEGs and some other files. So this is what it will look like when you pull it up. Again, the URLs will be as a part of the attachment with this presentation. So when you pull it up, this is what you'll see. You'll have the boundary layers. Currently, the state and county are selected, and you can go through and select a variety of boundary layers to look at. You can also click on the map, and it will tell you what provisional seed zone you're in. So here we're looking at the minus 10 to 15 degree minimum winter temperature, as well as the aridity from 3 to 6. The legend is here, though again, sometimes it can be challenging to figure out what shade of gray, green that you're looking at, and so clicking on the map is very useful. And again, this legend is just for the provisional seed zone maps, and you can look at it just for the Great Basin or the entire continental U.S. You can zoom in with this map as well as change the background imagery. So if you'd rather a topographic map, you can change that as well. And then you can also look at a variety of empirical seed zones, and to figure out what empirical seed zone you're in, you would need to click on the map. A word of caution, the map will show you everything that you click on, so you can make it a colorful nightmare if you click on everything. So do be sure before you generate your maps and print them out that you only have the items selected that you intend. And then finally, the most important aspect of this tool is the tools section, in which you can change the different boundaries you're looking at, the transparency of those layers, the provisional seed zones, as well as all of the empirical seed zones. Put in the lat long of the region you're working in. It will zoom in really close, so you'll have to come out and zoom out to the area you're working in. And then you can export the map as a PDF or JPEG and a variety of other file formats. And this will generate, you would need to scroll down on this page and you would generate a new page and be able to print it out for future use. This is my reminder for everyone collecting seed, whether you're the land manager, contracted seed collector, seed producer, to please always indicate the latitude and longitude or the coordinates with the datum from where you collected the seed. As we have more and more empirical seed zones developed, it would be great to know where those seeds came from. And the provisional seed zones may wind up getting divided up based on the empirical seed zone that's developed. So things will change. That is what science does, is we learn more and change with the knowledge we gain. So with this information, we'll be able to utilize those seeds that you've collected in the past. Okay, so the next step in the process of where do you get material? Now you know what seeds you need and where they need to come from. If you're in these regions, you can get forest seedlings through the Forest Seedling Network. They'll list all of the vendors that are available and tell you what they have available for purchase. For your native species, your herbs, your grasses, and forbs, you'll use the Native Seed Network. The Native Seed Network, you have a variety of options to go through. And you can click on your region and then even click on the eco-region. They are working on updating and doing a makeover for their website. So in the future, I'm told that they will have seed zone and seed zone information available. But at the moment, that is not present. You can also just click on buy seed or even search for seed that's for sale and put in the particular species you're interested in. And when you do that, you'll see the species and see how many pounds they think are available. And then you'll get this lovely table of what's available, and sometimes you get information of where the seed is actually from, and sometimes you don't. But the most useful thing here is the list of vendors that are available that have worked with these seed and have it available. And so you can go to their websites, and these are all links to their websites, and view what they have available and reach out to them, and then you can figure out what they have and if they have the seed zones that you need. In addition, if you're working with the Bureau of Land Management, you can access seed through their seed warehouse system. And if you're working with the Forest Service, you can contact Kayla at the Bend Seed Extractory. Kayla, if she has time, she may also be able to provide you with who may have seed that you may be interested in. So she may be a good resource for that. And then the nurseries that may be in your area will also be good to reach out to see what they have available. For instance, the Lucky Peak Nursery stores seed and grows plants for the national forests in Idaho, Utah, Nevada, Wyoming, Arizona, and New Mexico. And so I know there's a variety of Forest Service nurseries that provide seed for lands available. Other options are for you to go out and do it yourself or hire someone to do it for you. To do this, you'd want to collect at least five wild populations per seed zone per ecoregion. Andrea Kramer is talking next in this webinar series, and she may have more details on this, so I'd definitely tune into her webinar. You also want to verify that the area hasn't been previously seeded. Many areas have been seeded with cultivars, and so you do want to check what the previous history of the areas you're collecting from, because you don't want to collect something with the impression it's locally adapted when it just happens to have been a cultivar planted there in the past. You'll then contact your native seed company or extension service about increasing seed. You can chat with them about the area you're going to restore, the seeding rates and methods you plan on using, and then you'll be able to figure out how much seed you'll need to generate for the increase. You'll also want to consider your future needs and the viability of that seed in storage, because you may be able to generate enough seed for future use. So now I will go into the research that I've been working on. This is conjunction with Brad St. Clair at the Pacific Northwest Research Station in Corvallis, along with Nancy Shaw and Francis Kilkenny, who are at the Rocky Mountain Research Station in Boise. And we're working on testing the efficacy of seed zones for blue bunch wheatgrass. So here's the distribution of blue bunch wheatgrass. It is a long-lived perennial found throughout western US and Canada, and it occurs in a variety of habitats. So it ranges from 200 to 2,000 meters in elevation. Precipitation ranges from 21 to 187 centimeters per year, and the temperature ranges from 3 to 11 degrees Celsius. Brad St. Hilaire and his colleagues generated a seed zone map for blue bunch wheatgrass for the Intermountain West, and they divided this area into 11 different zones that are indicated here. And they ranked them from 1 to 7, and with some A's and B's in there occasionally for zones that seem similar, and then also incorporated the level 3 ecoregions. So these are the black lines, and the names are indicating the level 3 ecoregions for this area. So their suggestion is for a seed that you'd be transferring would be within the ecoregion and then within the seed zone within those ecoregions. The area that our experiment is focusing on is just a portion of this region, which we've zoomed into here. So now I'm going to explain our experimental design. So again, the seed zones would go from 1 to 7. 1 is indicating the warmer, drier climates, and the plants found here were much smaller. In comparison to those in the wetter, cooler climates, these were much larger plants. So our experiment has 2 transects, one in the north and one in the south. And within each transect, we have 8 sites in which we have planted a number of wild populations. So we went out and collected wild populations from each seed zone. So each seed zone has 4 to 5 wild populations that represent it, and then we plant these into each of the common gardens, and each common garden represents the environment of that seed zone. And this was then repeated again in the south. We established this experiment in fall of 2014. We put in seedlings. These seedlings were grown in a greenhouse, and then, so this way all of the plants were at the similar stage when we were putting them out, to then look at how do they survive and reproduce within each environment. We went back out in fall of 2000, or rather spring and summer of 2015, and collected survival, phenology, and reproduction data on all of these individuals. Overall, survival was really high in both the spring and the summer. So this is great for restoration, less so when looking at local adaptation. So if local is best, we would expect to see that the local population would always outperform, so fitness being number of seeds or survival, would always outperform the away site. So here I'm showing you each of the transects, so the northern transect and the southern transect, and then each of the common gardens going from the warm, dry common garden to the wet, cool common garden. And then we're looking at survival until spring. And all of the local seed zones are in the dark gray color, and the away seed zones are grouped together in the light gray color. And we can see that we didn't see that the local seed zones were statistically different based on the sites they were planted in. This could be for a number of factors. We did establish these plants in the greenhouse, so potentially they're still utilizing the resources they gained from the greenhouse. This is looking in the spring. This was only a couple months after we planted them in the ground. 2015 was a mild winter, and so there wouldn't have been a lot of selection due to cold hardiness since it was such a mild winter. And these are a long-lived perennial, so it may take a bit before we see a very strong signal, as has happened in a number of conifer species. Similar layout, but now looking at the summer reproductive phenology for each transect in each garden. Here we do see a significant difference in reproductive phenology by the local seed source based on the site they're planted. And the phenology is broken down to 0 was a category for vegetative reproduction, 1 was elongation, 2 was the grasses were in the boot phase and that the reproductive structures were starting to emerge, and then getting all the way to the seeds were maturing and dispersing would be 5. And so here we see that the away seed zone had a faster reproductive phenology than the local seed zone. This may be because these individuals are stressed and trying to hurry up and reproduce before they die, but this we won't find out until this year's data collection. However, in other sites we did see that the local seed zone performed faster than the away. And again, it could be because these are well suited and well adapted, and we'll be able to address this with our future data collection. And then finally, looking at reproductive stocks per plant. Again, looking at each transect and each seed zone, each site in the seed zone it's in. Here, there was a significant difference in reproduction based on the transects, so I had to analyze them separately. So I'm showing you the statistics for the northern transect and the southern transect. And we do see that the local seed zone performed differently based on where it was planted in comparison to the away seed zone. This particular seed zone in the northern transect had a really high water table, so our plants got very large and hence had a very large reproduction in comparison to all of our other sites. But we do see, particularly in the southern transect, that the local seed zone out-reproduced the away seed zone, and both are coolest and wettest, as well as driest and warmest seed zones. Again, noting that we may see more differences down the road since this is a long-lived perennial. In addition, we may still be seeing some residual effects of having life starting out in the greenhouse. Resources would have been acquired and these plants may still be subsisting on the resources they acquired while in the greenhouse. And so it may be until this year, until the second year of growth, that we will see a stronger response to selection since they'll be relying on the resources they acquired in this environment. Since we completed a reciprocal transplant experiment in that we moved all of the seed zones into all of the other seed zones, we will be able to compare how these are going to do in terms of climate change. And so this is showing you the climate for the area currently in comparison to the climate that is projected for 2050. And we'll be able to do projections to see how the climate will affect all of the traits. So here is showing you reproductive phenology and how that will change based on climate projections. And you can go to Francis's work to get more information on these predictions. Currently, we don't have the specific guidelines on how to utilize seed zones for climate change. That research is ongoing and we're trying to get to it as quickly as possible. But what I can say that if you're utilizing seeds that come from your seed zone, that they're going under similar pressures and have been under similar pressures, and so should be able to survive and do well with climate change. However, with our future research, we'll be able to address that more explicitly. I'd like to thank all of our cooperators. We wouldn't be able to do the research we're currently conducting without the help from private landowners and all of these federal and state agencies. In addition, I want to acknowledge all of our funding resources. Of course, without their help, we wouldn't be able to conduct this project. And this is one of our field sites here in the Santa Rosa Mountains in Nevada. And with that, I'd like to thank you for your time and attention and take any questions that you have. And I will leave up this URL, all the URLs for the sites that I noted, but these are also available in the handout that you have accessible with this webinar. Thank you.
Great, thanks so much, Holly. All right, so now we have time for questions. So if you have a question, please type it into your questions pane located at the top right of your screen. And we have a few questions. First one, well, actually the first one is a comment. It's from Courtney Rowe, and she said, "Thank you so much for highlighting her research."
My pleasure. Thank you for doing it, Courtney.
Okay, the next question, it kind of has two parts. So can you expand on what you mean by tree improvement and disease resistant? Is genetic engineering used, or is this truly native or natural?
Genetic engineering is not used. So with tree improvement, it's just artificial selection. So you would choose individuals that are growing faster for the growth rate or for disease resistance, have natural resistance to various fungi that they are encountering. And so those individuals are chosen then rather than those that are susceptible, and those are then propagated and then made available for restoration or reforestation.
Great, thank you. And then kind of a follow-up to that is, are there any rules that prohibit the use of GMOs?
There are rules that prohibit the use in terms of native restoration. I would say, A, they're not currently available, and I don't know of anyone that's even doing research to allow them to be currently available. But there are the basic rules that we would have to follow through the USDA and the EPA in following their guidelines before those seeds are even allowed to be available for commercial or public use. So we would follow those guidelines, but it is my understanding that no such material is in the works.
Okay, thank you. And you might have just answered this question, but it was from earlier, and it says, as climate change alters temperature and aridity, won't this undermine the use of seed zones as a seed selection tool?
Right, this tool won't be perfect forever, and we are in the process of incorporating climate change and how best to go about that. One of the tricky things is, the models are that they are models and they're not perfect, and we have as a nation and as a globe trying to change our patterns in carbon use, and so that may then change how climates are going to change and at what rate they're going to change, and all of that gets incorporated into models. And so as I stated, that forever these won't be perfect, but for the current use, they will allow us to put in more genetically appropriate material. One improvement that these are making is that currently a lot of very few propagated lines are put out there that come from one or two areas, and those are put everywhere regardless of climate. And so we're suggesting at least putting something that's locally adapted, which means they've already been under that selection from the recent change in climate, and should be able, and since those populations are persisting, they should be able to deal with current climates and short-term climate change.
Okay, great. And next question, is there information available for Hawaii seed zones?
Unfortunately, the provisional seed zone map was not made for Hawaii or Alaska. And I don't know off the top of my head for one that has been made, but I will look into that if you would like me to.
Okay, great. Will commercial seed supplies that rely on this information ever catch up with the science?
If the customers demand it, right? Customer demand, supply and demand, if this is what we want, just as we're seeing in our food stores that there's more and more organic food available, it's because what we're asking for, what we're demanding. Likewise, as land managers, if you say this is what I want and this is what I need, and you're not buying what they have, they're going to change. Mind you, we are working with seed producers and not saying you have to give us everything we want, and on the population level, we're trying to make this more economically feasible and not having, say, we need seed for every population, but for larger regions to restore.
Great, thanks. Several seed transfer zone studies have been conducted for grass species. However, fewer studies have been conducted for forbs. Our experience with conducting studies, STZ studies in U.S.
transfer.
Yeah, thank you. In U.S. Forest Service Region 1 is that forbs present challenges due to ectopic variation and specialized adaptation. Any thoughts on this? How do we develop cost-effective supplies of forbs across the larger geographic areas based on their specialized adaptation?
Yes, I mean, there's been some indication with that with the conifers, as I showed you the breeding zone map with Douglas fir. I think if anyone was to start off, they thought they could have done something more along the lines of western red cedar that just broke up a region into 4 different areas versus 16. And so we are going to see variation among species in how adaptive they are, how specialized they are. And I know there are forbs in the works that seed zone maps are being made for, and hopefully those will be available soon. But I know we need to come together with the data that we have and figure out, can we make, say, the next step provisional seed zone guide that may be specific to grasses or forbs, and try to break things down so that we don't have to make them for every single species, because that will be costly, but we still need to move forward and have some sort of guidelines so we're putting in locally adapted material.
Great, thank you. Is breeding structure implicit in the considerations of genetic variation when developing seed zones?
So by breeding structure, I'm making the assumption you mean if the plant is selfing or outcrossing.
Yes.
Okay, thank you. Is it implicit? No, I don't think that it is incorporated when making the seed zone guidelines. Again, it's going with the phenology, and so potentially the phenology itself may link that, may wind up separating groups if their phenology is quite different, but currently the breeding system or mating system isn't incorporated in how the seed zone maps are made.
Okay, great, thank you. How do folks working primarily on private lands access government supplies of public genetically appropriate seed stocks?
The plus side is a lot of our seed producers that do provide to federal government or federal lands are accessible to the public, so that Native Seed Network, all of the vendors on there can be accessed through the public. Some you would have to reach out. I don't know all of the operational rules for the BLM and for the Forest Service. So Kayla's information, she mentioned that she knows who owns the seed, so she may be able to give you that information, and you can ask them permission to the seed, and based on their restrictions, they may be able to give it to you. So I would just start with asking questions, and if it's available in your area.
Great, thank you so much. All right, that looks like the last question. Thank you all for your participation today. Oh, there is another question, so hold on.
Okay.
Pardon the complicated question. I'm not sure how to phrase this exactly. Do you foresee any possibilities for conducting rapid assays of sorts for species that we don't know the seed zones, perhaps at the family level, to predict what the seed zone might be?
Right. This is something we're contemplating and hoping to have a workshop to have some scientists come together and discuss this. As was noted, there have been a number of seed zones made for grasses, and some of which that are closely related. And even among those closely related, so not within the same genus, but different genus within the same family, they wind up having different seed zones. So we may have to, I think we need to just take the data that we have to get a sense if there is a rapid assay that we can kind of divide things up in another fashion. I think that is the next step for the provisional seed zones, is to go that route and see what additional information we can give without trying to generate empirical seed zones for every single species. But that work does need to happen and is actually currently on my mind.
All right, thanks so much. Okay, thanks again, everyone, and we would greatly appreciate it if you would take our 3-question survey of this webinar that will appear after the webinar has ended. I will post the recording of this webinar on our Great Basin Fire Science YouTube channel this afternoon, and the link will automatically be sent to you through the GoToWebinar system tomorrow. Our next webinar from Andrea Kramer that Holly mentioned will take place on Monday, the 8th, and it's titled Producing Native Plant Materials for Restoration, 10 Rules to Collect and Maintain Genetic Diversity. If you have further questions regarding this webinar or have requests for future webinars, please email or call me anytime. All right, again, thank you everyone for your participation today, and thank you so much, Holly, for your presentation.
Thank you for the opportunity.
All right, have a great day, everybody.
You too. Bye-bye.
Bye.